Good morning, everyone, or good afternoon, depending on where you are at in the world. So again, we're just going to talk a little bit about some of the initial characterization data of two of our new products. So I just want to go through a little bit of background about lysosomes. I think we all have a good understanding of what they are, but it's just good to refresh. So Christian de Duve was a scientist who discovered and named them, and for his research, he was awarded the Nobel Prize in 1974. So lysosomes are kind of the trash can of the cell. It's a cellular organelle that is membrane bound, and it is the site of extracellular substrate degradation and intercellular substrate degradation. That could include macromolecules, organelles, and substrates of that nature. Lysosomes contain a multitude of acidic hydrolytic enzymes, and lysosomes can vary greatly in size and density. Many of the new types of drugs now are being designed to be stable in the plasma, but then become activated once they reach the lysosome due to the unique characteristics of the lysosome being pH and the presence of certain catabolic enzymes. So this is a new field that people are developing, and we think that this is going to be a nice in vitro tool that certain groups will be able to use to help develop their products. And I also want to point out that lysosomes have isopycnic densities that are very similar to mitochondria, so purifying lysosomes from tissue is not a trivial process. Now this is just a very simplified cartoon of the endocytic lysosomal pathway. And for this particular example, I'm just using an ADC, an antibody drug conjugate, as an example of how this might move through the pathway. As the ADC binds a receptor, the receptor complex is in endocytosed and is trafficked to an early endosome. From there needs to be a trafficking decision made on whether or not the components of that early endosome are going to be shuttled back up to the plasma membrane or secreted through recycling endosomes, or if those complexes are going to continue down the endosome-lysosome pathway towards degradation. So if it is destined for degradation, that early endosome then fuses with a late endosome, and there's a pH drop. Now the pH drop is one of the unique characteristics of the lysosomal the pathway. Once that late endosome continues down the pathway, that complex and cargo is then shuttled to the lysosome where it fuses, and there's another pH drop. And once in the lysosome, that's where these complexes meet with a lot of the degradative enzymes, the catabolic enzymes that have been designed to activate the ADCs and release the warhead. Once the warhead is released, it then needs to be trafficked out of the lysosome through transporters, and it's from there you'd expect the warhead to interact with the target and then cause the phenotype that we would expect. Now there's also another side of the lysosomal pathway, and it's called the autophagosome. So autophagy is another pathway. This is the intracellular degradation pathway for substrates such as macromolecules and other organelles that are destined for degradation. So these organelles and cytoplasm get engulfed in an autophagosome. That autophagosome then travels and fuses with the lysosome where the contents are degraded. So with that little bit of background in mind, we just want to jump right into our rat tritosome product. So the name tritosome comes from combining two words, basically the triton, WR1339, which is now called tyloxapol. This is the reagent that we use to actually produce tritosomes. And then the second half of the word is obviously from lysosomes, because we are making modified lysosomes with triton. This method was first described in the 60s, and it was continuously developed over the years. as a way to increase the purity of lysosomal isolations due to overlapping densities of the two organelles in sucrose, which at the time was the most common density gradient material. There are newer materials now, however, they also have their own sets of pros and cons. So tioxopol or triton is actually trafficked to the lysosomes, sequestered there. It also causes changes in the lysosomal lipid composition. So the combination of the change in lipid composition and the sequestration of the tioxopol, we get a decrease in the isopycnic density of the lysosomes, which allows us to then use sucrose to purify and enrich lysosomes away from contaminating mitochondria. So this offers many benefits. We get highly purified lysosomes that are slightly modified with tioxopol. We get very high specific activity of common lysosomal enzymes such as acid phosphatase, cathepsin B, and acid RNases. And we believe that this is a simplified in vitro system that can speed up work due to the fact that the tritosomes that we prepare are disrupted and ready to use. We think that these will be more convenient in that there's no tissue culture preparation involved, there's no culturing of cells, and it's much simpler than a in vivo animal model. I do want to point out that all of our preps are EDTA and protease inhibitor free, so there's no compounds in there that may interfere with certain enzymatic activity. So I just want to quickly go through how we make our tritosomes. So we take a single dose of tioxopol, interperitoneal injection into a rat. 5 days later, we harvest the tissue, we homogenize it, and through various centrifugation steps, we end up with a pure preparation of tritosomes. So what we do is we layer the liver homogenate underneath the sucrose density gradient, and then through different centrifugation steps, they float away from the contaminating mitochondria and other membranes. And then we collect the band in the middle. So here's a representative isolation, if anyone's curious what it looks like. But more importantly, we get the question often, how do we know that these are pure? To address the purity question, what we've done is we've characterized the volumes of the sucrose gradients, the tritosomes, and we've looked for levels of enzymatic activity that we'd expect to find with the different organelles. So the cathepsin B is our enzymatic marker for lysosomes, and our cytochrome C oxidase, or COX, is the enzymatic marker for mitochondria. Now, as you can see, we get nice enrichment of cathepsin B in that white band that we associate with tritosomes. We have a nice increase there. If you go above or below that, you get a decrease of cathepsin B activity. Now, with that, we also get the concomitant increase or decrease of cytochrome C oxidase activity. The activity within the tritosome fraction is very low, whereas if you go just below that into the lower sucrose gradient, you'll see that that increases considerably. Now, this is what we'd expect, as we expect the tritosomes to float away from the mitochondria, so that we'd expect them in the more dense material, and that's what we see. So with this, our tritosomes have extremely high specific activity for your expected lysosomal enzymes, and these preps have a very low activity of enzymes that are associated with the mitochondria. So we feel like we have a nice purity and very high activity of our tritosomes. Now I want to switch gears a little bit, our human hepatic lysosomes. So just a quick outline of the protocol that we use for the lysosome isolation from human tissue. Here at XenoTech, we often get access to fresh human liver tissue. We go through a simple homogenation step where we make liver homogenate. We then take that liver homogenate, do a very quick ultracentrifugation spin, which makes our crude lysosomal fraction. That fraction is then run over a very low percent Percol gradient to further purify away contaminating membranes, and this leads us to our what we characterize as heavy membrane fraction. This heavy membrane fraction is then layered into an OptiPrep gradient, where then we spin overnight, and what we end up with is the picture that you see on the right, which is a density gradient with multiple bands that represent different organelles. And what we do to isolate the different fractions is that we basically start from the top and fraction top down, and we take 12 even fractions. And within those fractions, we then characterize specific lysosomal and mitochondrial enzymes for their activity to identify where in the gradient the lysosomes have banded to. And this is just a representative set of data from one isolation. What we can see here is that the cathepsin B is exactly where we expect it to be. The peak is around 6. Acid phosphatase peaks also in that same fraction. What's interesting is that the RNase is spread out over a couple more fractions, but it's relatively in the same area. And as our control is a cytochrome C oxidase, which is in the lower half of the fraction. There is a little bit of overlap between cathepsin B activity and cytochrome C oxidase activity within fraction 7. However, we would not consider that an acceptable amount of cytochrome C oxidase activity, so that would not be included within our fractions. So this is the activity based on units per mil. However, if we take the same data and we look at units per milligram, we kind of see a little bit different, but it still says the same thing. It's basically telling us that our lysosomes are fractionating to the top half of the gradient, while our mitochondria are fractionating to the bottom half. So we've also characterized these by Western blot. So we've done the enzymatic activity, and we looked at where in the gradient, LAMP1, which is a lysosomal membrane protein, where does that fractionate within the gradient? And we've also looked for COX-4 activity expression. So if you look at the first 3 lanes on the left, these are our 3 main fractions that we do on the front end before the gradient. So we have our liver homogenate, which is our starting material. We have our crude lysosomal fraction, which is after our first spin. And we have the heavy membrane, which is after our Percol spin to kind of clean up that crude fraction a little bit more. As you can see, with every manipulation, we get an increase of LAMP1 expression compared to the last. And these 3 lanes are normalized for protein content of 50 micrograms. Now, if you move on to the right side of the immunoblot, this is where we loaded our gradient. Left to right, it's top to bottom, and these particular lanes were loaded per volume, not per milligram protein. So we used 20 microliters of each fraction, and we ran it out and looked for LAMP1. So again, using a very traditional lysosomal marker, we can see that the presence of this marker correlates very nicely with the enzymatic activity data that we have on the previous slide. Also, the COX-4 activity coincides very nicely with our detection of that protein along the gradient. So this just confirms that our enzymatic activity does correlate well to the detection of these protein markers throughout their respective organelles. Another thing that we've done is we have taken the peak cathepsin B fraction, and we have assayed what proteolytic enzymes are present, and we've compared that to the liver homogenate, the initial liver homogenate, and we can see that we do get enrichment of specific lysosomal proteases, which also tell us that we were getting nice separation and enrichment. So if you look at the top, that's our starting point, and if you look at the middle panel, that is our purified lysosomes, or at least one of the fractions that contain them. And we can see an enrichment of cathepsin L and S, disintegrin, metalloproteinase domain containing protein 8 and 9, dipeptidylpeptidase 4, matrix metalloproteinase 8, urokinase, neprisilin, and presenilin. So again, this is just a nice comparison and confirmation that what we're isolating does appear to be lysosomes. And the saliva is just a nice control. It has a different assay readout, and it serves as a control. So I do want to take this time to point out that since we are using primary tissue from donors, there is inter-donor variability between the separation and the gradient. In most cases, what we see looks very much like the original data points that I showed you as far as enzymatic separation. However, there are occasions where we do see cytochrome C oxidase activity further up into the gradient. Now, we don't presently have criteria that would exclude this. However, we would look at very closely about not including this into the preps. So in conclusion, we have been able to purify human hepatic lysosomes, and on average they show a 2 to 5-fold increase in lysosomal enzymatic activity that you would associate with lysosomes compared to the initial liver homogenate. We see low contaminated activity from enzymes associated with the mitochondria, and we believe that this is going to serve as a good in vitro reagent with a human matrix that will complement rat tritosomes as an in vitro reagent for development. Now, I did mention that, as I pointed out with the immunoblot, fractions 1 through 6, those represent significant differences in densities. However, they all contain large amounts of LAMP1. So one of the questions that we're trying to address is, do these different organelles containing LAMP1 with their different densities, does that also represent different enzymatic activity? Does it potentially change the membrane composition? And then do those have significant effects on biological function? So what are the potential current uses of these two products? Well, we feel that it's going to be a good in vitro diagnostic tool to conveniently and quickly evaluate potential changes in lysosomal stability due to targeted modifications of the biopharmaceutical. And we think that with that data, it will help narrow and direct development tracks of the biopharmaceutical. And we think that the people that are going to want to use these are those that are looking at ADCs, siRNA or RNAi type technologies, biodegradable copolymers as delivery mechanisms, and potentially nanoparticles. Now, in this particular presentation, we've talked a lot about drug development, but there's other things outside of that box where these particular reagents could be used, and one of those just may be the cosmetic industry and microparticles. They have recently been banned for use because they're not biodegradable. They're not being degraded, and they're accumulating in certain organisms out in nature. So there's potentially more uses for these types of reagents than what we presented here.